Well, welcome to a brand new start. As we begin officially the second half of the course regarding lecture, you've already been through Lab 6, which is the nervous system. Today we'll be discussing the nervous system. This is going to cover for us 4 chapters in the textbook, 11, 12, 13, and 14. So we'll be spending some time here. 11 will be an introduction to the nervous system and the parts of the neuron. 12 will discuss the spinal cord. 13 will discuss the brain, and 14 will be about the autonomic nervous system. Now, I don't know yet, but I might have to record the chapter 14 autonomic nervous system lecture in advance. And the reason is this. I have as many minutes to instruct you in the fall as I do in the summer. The problem is when we get together for an exam, it's an entire 2-hour and 10-minute period that I'm giving up for an exam, whereas in the fall or in the winter, I only have an hour and a half class. So I'm losing 40 minutes of instructional time every time we have an exam. And I've now lost about an hour and a half of instructional time between the 3 exams. So I likely will have to pre-record and post on YouTube a lecture. It'll take about an hour or so over chapter 14 on the autonomic nervous system. Just anticipate it. I anticipate having to do that, and I'll send out the email when that time happens. But that chapter wouldn't even make sense necessarily until we get through the next 3 chapters. So hang in on that. Just know that it's likely coming your way. The next exam, again, exam number 4, will all be about the nervous system, chapters, again, 11, 12, 13, 14, and with that, vocabulary what? What's our syllabus tells us that vocab 51 through 70. So we'll have 20 slides to look at for vocabulary, and that exam will be when?
7th.
The 7th of July. So you'll get to go away, watch the fireworks, but in between the fireworks, you'll have your notes out, right? And you'll be looking over that weekend. I know that's sometimes a tough weekend if you're going away for family plans and things like that. So again, anticipate that. Try to learn as much as you can as we approach that holiday weekend, and then when you come back, enjoy yourself, and then come back and review. But please don't wait until after July 4th to begin your serious study of this material. This next exam is traditionally it's the longest exam. I'll tell you that right now, that the 4th exam is the longest of the semester exams, but you do have the 2-hour block of period to take it, so time won't be an issue for you, I'm confident. So let's get started with the vocabulary, going back to that. And how many lectures do I have with you? Count them up between now and the exam. So today, Wednesday, and then Monday and Wednesday, and then
7, 4, just 3.
3 lectures, right?
Mm-hmm.
Okay, so I've got to go through about 7 slides then for your vocabulary each time. So let me go through the next 7 slides with you, and we'll do 7 the next 2 times. So starting with slide number 50, we've seen this term already. Sarcolemma was the outer layer of a muscle cell. In lab, on the neuron model, the term axolemma came up. That is the outer layer of the axon. So lemma meaning a rind or a peel. Lepsy, a seizure. Yeah.
We went through let on the last.
We went through yep. So I'll start here on my slides and it'll be okay. So we'll start here. So lepsy, again, epilepsy, I remember going through this. So seizure upon seizure upon seizure, right? And let, small. And so platelets are little tiny fragments that we'll look at when we get to blood clotting. And then, of course, leuco, white. There are the white blood cells called the leukocytes. Lig, you know a ligament, right? A ligament is a structure that connects a bone to a bone or some other structure to another structure. So to tie or to bind. The lipids are fats. Ology or logy is a study of something. Here we are in a biology class studying something about life. And lysis, the breaking down of something or dissolving. So hemolysis or hemolysis is the breaking down of what? Hemo, blood, right? Hemolysis. Glycolysis would be the breakdown of sugars, glyco. Lateral, you know, means to the side. So lateral, side. There's leuco again. I don't know why it shows up twice. So clearly it must be important. Lith, stone. If you have kidney stones, they might put you in a lithotripsy, which is basically a sonication type thing that will try to break down the stone. So maybe you've heard of a lithotripsy. It's a breaking down a gallstone or a kidney stone. And then lucid. If grandma's still lucid, her mind is still what? With it, clear, right? So lucid or light, we'll hear that term used.
I don't think lat's on our stuff.
Then put it on. Okay. So lucid, light, or clear. You've already seen the stratum lucidum, that light layer that was found only in thick skin.
I see it now.
Yep. There's just what happens is every semester I add a few more terms to this or take a few out, and every once in a while something changes, and I can't seem to find your version on my PowerPoint. So I'll keep looking. There hasn't been too many changes. You know lunar meaning the moon. There are valves in your heart called the semi-lunar valves. They sort of resemble half a moon structure. We'll see those when we get to the heart. Lyso, break. Remember, lysosomes were the vesicles that were able to break down substances. Macro meaning large. There are large cells called macrophages in your blood. And macule. If you've heard of someone having macular degeneration, it's a degenerative eye condition. And what begins as a spot in their vision gets larger and larger until they lose their ability to see. So macular degeneration, a spot or a stain in their visual field. Malaria. Before we knew what caused malaria, they thought it was caused by mal-air, bad air. They didn't know what it was, right? So it kept the name of it. So mal meaning bad, malnutrition, malicious, you name it, right? Mal means bad. And area literally meant we didn't know what caused malaria. We thought it was just bad air. Malacia, softening. Osteomalacia, a softening of bone, for example. Not a good thing if you have soft bones. Mam and mast, both referring to breasts. Think mammography or a mammogram or mastectomy. You've seen the term meatus before. A meate, a tube-like passageway. We saw it in the external auditory meatus of the temporal bone. And medi, toward the middle. So something that's medial, you know, is going toward the middle of the structure. If you win the lottery, right, the mega, whatever it is, right? So mega, really large. There are cells called megakaryocytes. You tell me what megakaryocytes would mean. Mega meaning large. What is karyo? Nucleus. Yeah, nucleus. So megakaryocytes would be cells with a very large nucleus. And we'll see those when we get to the blood. Megaly, an enlargement. Splenomegaly would be an enlarged spleen, right? Anything with megaly in it is an enlargement. Melano, that is a typo. It should be M-E-L-A-N-O. Melanocytes, we've seen melanocytes. They were the pigment-producing cells in the skin. They produce melanin, which was a darker pigment, but melano, from its derivation, actually means black.
Isn't splenomegaly spelled wrong?
Yeah, there shouldn't be an H, right?
Right.
Splenomegaly, there should not be an H. Good catch. Mens and menstru, monthly. So the menstrual cycle, the monthly gift. Amenorrhea, right, would be an absence of monthly flow. Rhea, we'll see later on, means flow, like diarrhea or rhinorrhea. Meres and mers, they both mean parts. We saw the term sarcomere. It was the unit of muscle that moves at the microscopic level. And you've all heard of a polymer or a monomer, a molecule made up of many parts or one part. Mes in the middle. When we get to the digestive system, we'll see that there's a layer of very thin tissue called the mesenteries, and they're sort of in the middle of the intestines and kind of bind it all together and hold it together. Meta, beyond or after metastasis, right? Cancer cells are known to metastasize, that is, they're to go beyond their normal limit. They don't stay still, they spread to other parts of the body. And metabol, change. Your metabolism or all the chemical reactions, all the chemical changes occurring in your body. So metabolism, all of those changes collectively. And then I'll finish up with 57 today. METR. If you see this in the root, for example, if you look at endometrium, endo meaning within, METR referring to the uterus or the womb. So it's a layer of tissue within the inside layer of the uterus. We'll deal with that when we get to the reproductive systems at the end. Micro, you know that one means small. Mitosis or mito. back when scientists were first looking under the microscope and they saw these string-like structures when cells were dividing, right? They didn't know what they were. They didn't know they were chromosomes. They didn't know what this was. But they saw during mitosis these thread-like chromosomes. And so the derivation of this term mitosis comes from the fact that you can see these thread-like structures during cell division. And then finally, mono meaning one or single. So a monomer, something like that. So that'll get us through 7. And we're going to be going through, was it 70? Phleb, right? Or is it 51 through 70, right? So we'll go through Phleb. So you can go ahead and make up those cards and carry those with you. And again, during the fireworks, you can be flipping through your vocab cards. Everyone around you will be really impressed of how studious you are. Okay? I used to go to football games at the University of Florida and carry books with me. I mean, I wasn't the only one, right? So in between, in a boring play, pull out the book, pull out the notes. So you'll adapt. You'll get used to carrying around your notes or something with you most everywhere you go as a full-time student. So let's begin this journey on the nervous system. The nervous system, pretty complex. And my job will be to break it down into a meaningful discussion with you. There are quite a few structures that you've seen in lab already. During this unit, we'll be looking at the parts of the neuron. We'll be looking at different nerves of the body. We'll be looking at the brain and the spinal cord structures. We'll also be looking later on at the eye and the ear. Now, the eye and the ear will not be part of this exam. They'll be part of the next exam. So we're going to be with the nervous system for a while here in the course. But just know we're going to be going through many of the structures that you saw, or many of the models that you saw in lab, will begin to make a lot more sense to you than perhaps they did the first time around in class, in lab. So some of this you have gleaned from lab. So you know that the nervous system is the body's communication system. It's one of two. The other major communication system is the endocrine system, right? All the hormones that are traveling through our blood. But we're going to be dealing first with the nervous system. And we can divide the nervous system up in a couple of different ways. One is by just structural or anatomical divisions, the other by function, and both of them were already introduced to you in lab. We'll start off by thinking about the structural or the anatomical organization. And this is what you saw in lab. That is, we can break it down into the central nervous system, which includes just the brain and the spinal cord, and additionally, everything outside of the brain or the spinal cord is in the periphery. So we say it's part of the peripheral nervous system, and this is going to include the nerves of the body. In chapter 13, I'll be discussing cranial nerves. There are 12 nerves that pop out of your brain, basically, up in the cranium, that are going to be going to control your vision and your hearing and your taste. We'll talk about those in a couple of chapters. You were introduced in lab to 31 pairs of spinal nerves. Those are those nerves coming off the spinal cord, right? Those 31 nerves, and we'll talk more about those in the next chapter. And there are also these structures called ganglia, which are clusters of cell bodies or clusters of somas that are located outside of the CNS. Now, that definition of a ganglia will come back later on. It doesn't make sense to you quite yet. So looking at this from a picture standpoint, we've got the central nervous system shown basically in the tan, the brain. And remember from lab that the spinal cord only goes down where? To about L1, right? And that's about as far as the spinal cord goes, and that's at vertebra L1. Below that, there's still nerves traveling down through the vertebral foramina, aren't there? But those clusters of nerves are collectively called the cauda equina, and we'll discuss that more in a moment. Everything that is outside of the brain or the spinal cord is instead referred to as part of the peripheral nervous system. And again, this is going to include the cranial nerves, the spinal nerves, and the ganglia. And the ganglia we'll come back to it, but the ganglia on this image are all these little round dots that you're seeing next to the spine. So these little round dots that you're seeing is what it's pointing to as ganglia. Hold on, and we'll discuss that later. When you're looking at this image, you can see nerves that you're beginning to understand naming. So what is this nerve coming down the front of the leg here? Remember what that nerve was? Coming down the front of the femur is the femoral nerve. And what nerve would be coming down along the pinky? What bone comes along the pinky? Ulna, so that's the ulnar nerve. What nerve is coming down along the thumb side? The radial nerve. And right down the middle, running out of colors here, but right down the middle would be right down the middle, oh, that's a big line. But right down the middle between the ulnar and the radial is the median nerve. Okay, so the names of these nerves really aren't that tough. There's a couple in there that are kind of new and unusual, but many of these nerve names you're already familiar with because they're named for either the bone they travel through or the part of the body to which they travel. So that is the anatomical way of breaking up the nervous system. We can also break it up in a functional way. And that is, there are certain neurons that are collecting the information. You know those as the what? From lab, what classification of nerves are receiving or collecting the information? Sensory, right? And the other name for the sensory neurons were the afferent. Right, so the afferent or the afferent, I'll always overemphasize the A. So the afferent neurons are the ones that are sensory, that are collecting the information. Where is this information coming from? Either the external environment or from your internal environment, right, from inside your body. Then there are some neurons that are receiving and they're processing and evaluating this data. You know those as what? They were the in-between neurons. What's the word for between? The interneurons, right? The interneurons were the neurons between the incoming and outgoing. They were the processors, the thinkers, the integrators. And then finally there are neurons that are going to respond, or that is dictate something. And those are the what? Efferent or motor. Right, so the motor neurons or the efferent neurons, and where are they sending their information to? Either muscles, right, telling the muscle to move. If it's a skeletal muscle, it's telling your body to move. If it's the cardiac muscle, it's telling the heart to pump. If it is going to smooth muscle, it's telling your gut to contract. Or these motor neurons can be going to glands, telling those glands to secrete something. Right, so we're starting to get this overarching idea of the nervous system. So I've already said this, I believe, but we'll say it again. There's the sensory side of the nervous system. Also the afferent, as we already said, these are the neurons receiving the information. This is again coming from your external environment: vision, hearing, smell, taste, touch. All of your quote senses, right, are being. Those sensations are being gathered and sent to your brain through the sensory neurons. And remember what afferent means, right? AF means what? What does AF mean? Remember, AF means toward, right? And ferent, to carry. So we're carrying toward what? Carrying toward the brain, carrying toward the spinal cord information. And then efferent, right? EF means away from. So we're carrying things away from the spine or brain and carrying things out back to the periphery. Now what you'll see is that there's a combination here of both the peripheral and the central nervous system, right? Because the motor neurons are peripheral, the sensory neurons are peripheral, and the interneurons are what? Central. Let me say that again. The incoming sensory neurons are part of your peripheral nervous system. The interneurons that are collecting and processing the data are part of your central nervous system. and then the motor neurons leaving are once again part of the peripheral nervous system. So we need both of these portions for an overall working nervous system. I like this particular table because it kind of breaks everything down and gives us a little bit more detail than what we've discussed so far. So again, this is all about the nervous system collectively. We know these two portions. We know we can break it up into the sensory side, the afferent side, and we can also pick it up or divide it up into the motor side or the efferent side. But let's break it down even one more step. So over here on the sensory side, there's somatic sensory and visceral sensory. What do those words mean? What does somatic mean? Body, right? So this is the sensory portion coming from essentially your external body, your soma, from your body. So this is going to include information coming to your brain from your skin, your joints, your muscles, your eye, your ear, your taste buds, right? Everything to do with your special senses. Then we know also that incoming information is not coming just from your external, but also from your internal structures. That's why it's called the visceral sensory. So these are the signals coming in, for example, from your stomach, from your bladder. You may not be thinking about it, or maybe you are. Are you hungry? Is your stomach empty? Is it growling? Right? All that information, is your bladder full? Is your bladder empty? All of that kind of information is also going back to your brain right now, maybe completely unknowing to you. But there's always an all systems check going on in your body from these sensory neurons sending signals to your brain. On the motor side, we can also break it up into two parts. Somatic motor. So again, if soma means body, the somatic motor must be what? Neurons that move the body. And that's exactly right. Sometimes this is called the voluntary side because this is remember your skeletal muscle is voluntary muscle. And so if you're voluntarily moving your body, you are using somatic motor neurons or part of the nervous system on this side. If instead you're sending signals to your involuntary muscles, which include what? Involuntary muscles include your cardiac, heart, and the smooth. Or if you're sending signals to a gland, those signals are part of your autonomic motor. Now you're going to say autonomic, you're going to spell autonomic, but you're going to think in your head automatic. So the autonomic portion are the things that are happening for you, quote, automatically. You are not thinking about squeezing your intestines for digestion. You're not thinking about causing your heart to beat. And you're not thinking about telling your pancreas to secrete gut enzymes and gut juices, right? Those are all happening without your voluntary control. So we say that's your autonomic motor or your visceral side or your involuntary side. Once again, I showed it to you in table. Here it is in words. If you didn't catch it all, there is an extra word on here, though, that I want us to focus on. So again, we have the sensory side or the afferent side of the nervous system. We broke it down in that table into somatic sensory and visceral sensory. There's a word in here, though, that I want to introduce to you that I want to make sure you leave here understanding. So let's think about this again. Sensory, soma. Sensations from the body, more from the surface or from the outside of the body. So that's going to include your touch, your pain, pressure to the body, vibration on the surface, temperature changes. But here's the word I want you to focus on. This is a brand new word for you. Proprioception. Proprioception. Now, proprio means self. Self. So what this is an awareness of self. Proprioception is an awareness of your own body position. Are you able to close your eyes and tell if your arms are bent or straight? Right? You know if your arm is bent or straight. You don't have to look at it. You don't have to look in the mirror. Why is that? You have signals coming from your joints and from your muscles that are reporting to your brain what position your body is in, right? And that is your sense of proprioception. Okay, very nice word to know, proprioception. Then again, we can have your visceral sensory, like I said, your viscera. You are now, your bladder, your stomach, your heart, your lungs, all of your organs are sending signals back. Those are your internal organs, sending signals back to your brain as well. On the motor side, again, mostly already discussed, we've got the somatic motor. This is definitely your voluntary portion. This is only going to what kind of muscle? Skeletal, right? Your voluntary type of muscle. Versus the autonomic motor, which is going to all those things which are involuntary, which you know include your smooth muscle, your cardiac muscle, and your glandular secretions. Now, we're going to be spending some time, I need to update this number here, not chapter 15, but chapter 14. And when the chapter I was discussing that I probably will have to do a pre-recorded lecture and have you listen to, will be all about the autonomic nervous system. There's actually another whole division of your nervous system that we'll wait to the end and talk about. And this is broken down into terms you probably already heard of, sympathetic and parasympathetic. Sympathetic is your fight or flight response, and parasympathetic is your rest and digest response. And we'll talk more about that later on.
You have 15 and you're making it 14, but on ours it says 18. It really should be 14. Ours says
Your notes say it should
Ours
be
say
14.
18.
14. 14. So sympathetic and parasympathetic will be chapter 14. Okay. So I can go back then, and on this table, go ahead and underneath autonomic motor, you can put sympathetic and parasympathetic. Okay? And we'll break those down more. But just know that that's going to be our discussion in chapter 14. So the autonomic motor can be further broken down into sympathetic and parasympathetic. So that gives you an overarching view of the nervous system and its different components. Now we're going to put this whole thing together and figure out how it works. Now, when you think of a cell, what's the first thing that comes to mind? Popcorn out some ideas about cells. When I think of a cell, I think of something that's spherical, right? Round, if you will. And I know that it has a nucleus, and I know that it has different organelles, like the mitochondria and the Golgi and the rough ER and the ribosomes. So the first thing I think about for a cell is some spherical round cell. And then we saw, epithelial cells can be cuboidal or columnar, so we kind of stretch our ideas of a cell. And then we saw in the muscle unit that muscle cells are, wow, really, really different. Muscle cells can be a foot long. Muscle cells can be 18 inches long. Think about your sartorius muscle. That's a pretty long muscle. And muscle cells are as long as a muscle itself is. Neurons are the nerve cells, and they're not the only kind of cell in your nervous system, but they're the primary one we're going to talk about. Because the neuron is the kind of cell that sends the electrical signal through it and allows us to communicate with our systems so very quickly. So we'll be talking about neurons, and then we'll also be talking about glial cells. What did we learn about the word glia? Yeah. So glia meant glue. And we'll see that there are other cells in the nervous system that are sort of gluing everything together, these helper or glial cells. So let's go back first and look at neurons. And you've seen a neuron in lab, and we're going to label and learn mostly about the neuron. Now, what's unique about the neuron is that it's excitable. And that's a fancy way of saying that it can carry signals and it can respond to stimuli. And three characteristics about the neurons. Number one, they're highly metabolic. What does that tell you? Yeah, they have a lot of metabolism, a lot of chemical reactions going on. And that also tells you that you require a lot of energy. right? So there's a lot of metabolism, a lot of chemical reactions going on. They're very ATP needy, lots of energy needed. Therefore, what organelle would you find a lot of in neurons? Mitochondria, right? Just like in muscle, very needy, also so are neurons. Neurons are also have extreme longevity. Basically, once they're formed in you as an embryo, that's it. They're going to be with you the rest of your life. And if you damage a neuron, they cannot, number 3, divide. Neurons are not mitotic. They cannot divide. So once they're formed, they cannot divide and replace themselves. Also, once damaged, right? So that's why spinal cord injury is so devastating. Because once we have a brain injury or a spinal cord injury, the neurons don't regenerate. You've lost that portion of your nervous system, and whatever connections were made by those neurons is lost. There's a little bit of wiggle room in that. But for the most part, lose a neuron, it's over with. And so we look at those 3 characteristics: highly metabolic, needing lots of energy, longevity, and non-mitotic. One of the reasons that neurons are not mitotic is they lack what organelle? I know this came up in some of your mastering questions. Yeah, neurons lack what? Centrioles, right? And what do centrioles do? Centrioles were that structure that, like Spider-Man, right? The mitotic spindles kind of spit out, and those were necessary for the chromosomes to be pulled apart during mitosis. So without centrioles, you can't divide, you can't do mitosis. So these cells, even if they wanted to, can't divide and replace themselves. Versus the glial cells, the other cells, the glue cells of the nervous system. Now the glial cells are really, I think of them as being like the helper bees. Right? So you got the queen bee, that's like the neuron, long-lived, protected, and then you have all these drones and helpers. I may be using some of the wrong terms, right? But all these other busy bees around it. And so that's what the glial cells are. They're there to support and protect the neuron. And they are not excitable. So they do not send electrical signals, but they are replaceable. So they are mitotic, right? So they're shorter lived. They are replaceable. If they get damaged, they can be replaced. There are 4 types of glial cells. We'll look at each of them after we go through the neuron. But there are astrocytes, ependymal cells, microglia, and oligodendrocytes. And we'll look at each of those. I think all 4 of those were mentioned in that neuron diorama that you saw in the lab this week, and it made no sense to you at that point. But now you'll be able to go back and say, oh, now I get it. I see which ones of those are astrocytes, which are microglia, etc. So let's start off with the neuron. This was introduced to you in lab a little bit. And neurons are the longest cells in your body. The axon, the long process, can be as long as a meter in a person my height, with neurons going from my spinal cord way up at L1 all the way down to my little toe. So going the entire length are some single cells called the neurons. And let's talk about the different shapes and sizes. But first, let's get through the basic anatomy of a neuron. It's no different than any other cell. It has a body, which we refer to as a soma, the cell body or the soma. And this is the center, if you will, of the cell's activities. And then within the soma, within the cytoplasm of the soma, there are going to be Nissl bodies. Now we're stuck with the name of the person who described it. So Nissl, N-I-S-S-L, that's why it's capitalized. And the Nissl bodies, when Nissl looked under the microscope and looked at neurons, what he noted is that the cytoplasm seemed to be filled with these dark staining globs. He didn't know what they were. So, those are mine, right? I call them Nissl bodies. I'm the first one to describe them. Now what we know them to be are clusters of ribosomes. This neurons are so metabolically active and are so busy making proteins that there's such a dense collection of ribosomes and endoplasmic reticulum that you can actually see them under the light microscope. Not individually, but you can see evidence of these clumpy-like structures called Nissl bodies. And another term that I want you to pick up on is perikaryon. Break that word down for me. We haven't had peri yet, but you know what perimeter means. Peri means around, right? The perimesium went around the fascicle. So peri, carry-on. Caryo, nucleus. So what must this be? The perikaryon is everything around the nucleus. You would call that the what? Cytoplasm. So again, we have some new vocabulary for a rather unique cell. So the perikaryon, another term for the cytoplasm of a neuron. Then, as you saw in lab, there are dendrites, and we know what that word means. Breaks down. Dend means tree. Ite means little. Most cartoons are going to show you a neuron with maybe 5 or 6 or 10 dendrites. In reality, some cells in your body may have 10,000, as many as 100,000 dendrites on it. So this is a huge, huge number of these processes that stick out of the soma. And then finally there is going to be axons, and I probably should make that singular because there's just one axon per neuron. So whereas there can be many, many, many dendrites, there will only be one axon, and that axon will be longer, and we'll discuss how to identify it. So let's start off with dendrites. Again, we know they mean little trees. They're shorter, smaller processes. Remember, a process in bone was anything that sticks out. Same is true here. These are small processes. And like I said, neurons usually have more than one or many, many dendrites. It is possible for some neurons to only have one dendrite, but most are going to have many. And the dendrites are receiving the electrical signal. So the information traveling through an axon, through a neuron is always unidirectional. That is, the movement of electrical activity goes in one direction. And this information comes in at many of the dendrites. Think about this. The more dendrites that a neuron has, the more information that one neuron can receive at any one time. So you're going to find that the more dendrites a neuron has, the more it's kind of in the center of your nervous system. It's going to be a receiver of many signals. So it's like a manager, right, who has many, many underlings who is working for him or her, and so they've got their tentacles out, if you will, to many, many people. Now that's in comparison to the axon. There will only be one axon. It's typically much, much longer. It's going to be the longest of the processes. There's a term here that I want you to hang on to, and that is nerve fiber. Just to confuse us. In tissue culture, or in connective tissues, what were fibers again? Elastin and collagen and reticular, they were proteins. In muscle, if you said muscle fiber, a muscle fiber was the entire muscle cell. Okay, one more permutation here. A nerve fiber equals an axon. Not the whole neuron, but just the axon. So when you hear about the term nerve fiber, it's referring just to the axon portion. Now, most neurons, all the neurons that we're going to deal with in this class are going to have a single axon. And down that axon, the nerve impulse or the electrical signal is always traveling away from the soma. So axon away, A away. The axon is always carrying the electrical signal away from the soma, away from the cell body. And where's it going? It's going down the axon. And what's on the other end of the axon? Could be a muscle, telling that muscle to contract. It could be a gland, telling the gland to secrete. It could be another neuron, informing that next neuron of what's going on. So we'll see that the axon is the part that reaches out to another portion of the body. The axon connects to the cell body at a region called the axon hillock. And the axon hillock is sort of a triangular, another way of thinking about it is a conical shape or a funnel-shaped region. Okay, a conical or funnel-shaped region, and you'll see that in the diagram coming up. It's a funny little word, the axon hillock. There can be branches of the axon, and those are going to be simply called collaterals, right, off to the side, lateral. And at the very, very end of the axon, as I described in lab, there are little branches. I used 3 different words for those branches at the end of an axon. I said they could be called axon terminals. They also could be referred to as telodendria. Here's that word. And in lab, the image on the screen said instead what? Axon terminals equal telodendria equal terminal arborizations. Remember, trees at the end, little branches at the end of the axon. And then when you get to the very, very end of the axon, you have these little swollen areas called synaptic knobs. So, very similar picture to what you've seen. All of these are what coming in? These are all dendrites, little trees. There is an arrow here, right, that tells you that the information is traveling inward. So typically dendrites are receiving the information, the electrical information, the stimulation is coming in through the dendrites. Then here you can appreciate there's a conical shaped or funnel shaped or a triangular shaped region. This is the axon hillock. And attached to that axon hillock is the axon. It tells us here that, in this arrow, that the direction of movement again is away from the soma, traveling down the axon, away from the soma toward the axon terminals, which are then found way down here at the bottom. So they are the telodendria. Again, they are also called the terminal arborizations, weren't they? Terminal arborizations. And at the very, very end are these little bulges called the synaptic knobs. Along the length of the axon, you're appreciating how long these axons can be. And I've also told you that once a neuron is damaged, it can't replace itself very easily. So we better protect these axons. So there are other cells that wrap themselves around, and here's one, and here's one, and here's one. And those cells are the Schwann cells. Those Schwann cells are protecting the axon from damage, but the Schwann cells are producing a protein called myelin. And myelin is a white-colored, shiny, fatty type protein, what we call a lipoprotein. And this protein is protecting the axon. It's also helping to insulate it, almost like little strips of insulation tape along a wire. Those Schwann cells can be replaced, right? They're helper cells. So they have their own little nucleus, they have their own little metabolism, and if they get damaged, they can be replaced. But the neuron cannot. Last thing I want to show you on this particular slide is that in between the Schwann cells are gaps, and those were described by a guy named Ranvier. So they're called the nodes of Ranvier, these little gaps in between the axons. Oh, there's one more term on here we might as well throw out while we're here. Neurolemocyte. It says that a Schwann cell is the same as a neurolemocyte. Break it down for me. A cell site that lemma, rind or peel, goes around the neuro. In this case, the axon. Okay? So it's a cell that wraps itself around like a rind or a peel. So you'll see the word Schwann cell named after the guy who characterized them, and you might see the term neurolemocyte instead, an equivalent term for these Schwann cells that make the myelin. So you're going to know and love the neuron, right? It's just a lovely cell.
Is the Schwann
So the whole, so yeah, I'll show you a better picture in a moment, but this entire thing is a Schwann cell. And the Schwann cell does make layer after layer after layer of myelin. And what you see is that each of these cells has its own little purple nucleus. See it? So each of these cells has its own metabolism, has its own set of organelles, but its primary job is to make myelin, hundreds of layers of myelin, creating this electrical tape layer, layer, layer. Now, under the microscope, I wish we had any slides that look this good under the microscope. But what we're able to see here, dendrite, 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 and then it's kind of a guess, but because in this particular view, I'm able to see that much larger triangular region called the axon hillock, and because I'm lucky enough in this view to see a much longer structure here, we're going to call this the axon. Now, can I tell the axon for sure? No. But I'm imagining, right, that if I cut this neuron from a different view, that it's possible. I'm not saying they're wrong, but I'm just saying there's no guarantee that perhaps this, maybe this axon ends right here, right beyond the view here, and in reality, this guy goes on for 10 yards, right? And this is actually the axon hillock. From a different view, it might look more convincing. Again, you cannot tell a dendrite from an axon in a view quite like this unless you could see for sure the length of the axon compared to the other dendrites. I'm just telling you that to realize that it's not quite as simple as it looks, but they did label that as the axon, so we'll call this the axon. Now, this would be the what? All this would be the soma, and this would be the nucleus, and I can see some dots out here. They're not the most obvious thing, but all those dots are what? Nisobodies. Oh, don't you love it? They're also called the chromatophilic substances. Now, again, what's chromo mean? Chromo? Color? Philic. We haven't been there yet, but a philia or a hydrophilic, an affinity for or love of. So what does this tell me? That whatever this stuff is, these little dots, they have an affinity for color, right? Okay, so chromatophilic substances, don't throw tomatoes at me, I don't make this stuff up, right? I'm just a messenger. So the chromatophilic substance, AKA the Nisobodies. And what they are, in reality, are what? Clusters of ribosomes and endoplasmic reticulum so densely packed within the perikaryon in the cytoplasm that we can see it under light microscopy.
Is that what those other dots are around it? Like
Ah, no.
dendrites?
Good point. Now the other dots, all these other dots represent nuclei of other cells. You tell me, what kind of cells are they? Some variety or variation of the glial cells. Right, so again, you got Mama B here, you got the big queen, she's the most important cell, the neuron, and around her you see a plethora of smaller glial cells of different types. So that's what all those other dark nuclei are. Okay. Now, neurons come in different shapes. The study of shape would be what? Here's a term we haven't quite gotten to, I don't believe. Morph. Shape, right? So morphology would be the study of shape. If I ask you what is the morphology of a neuron, I'm asking you what is the shape of that neuron. And we're going to talk about neurons being of different shapes and of different locations. And largely, their function will be based upon where they're found. Let me first go through some morphology, some shape differences of neurons. And the names make, I think, total sense. Some neurons are said to be unipolar. Now, you may recall in lab, I had you fill in that table, and I said, oh, in that table, put unipolar, and other tables put multipolar. So that will make more sense to you now as we go through this. When you hear the word polar, what comes to mind? Besides polar bears. If I said that cells have polarity, right? They have different sides, like north and south pole. And a pole would also be sort of like something sticking out. So a neuron that is unipolar would only have how many poles? One. In other words, one process that sticks out of it. What would a bipolar neuron have? Two. And a multipolar? Many. Okay? Now the poles, you can almost think of them as being wooden poles, something sticking out of the cell. I'll show you these in a moment. You may recall that in lab, I told you that sensory neurons were unipolar. So there's that connection right there. I want you to know that your sensory neurons are, their shape is going to be described as being unipolar. Whereas your, let me add this term, whereas your interneurons and your motor neurons, they are multipolar. Okay, so the interneurons and the motor neurons are multipolar, which tells you what? Many, many processes, which means what? How many axons? Always and only one axon. So that means that there's one axon in many dendrites, multipolar. There are some neurons in your body called bipolar neurons. We won't be discussing them anymore this semester, but what would they have? Two poles, one dendrite, one axon on opposite sides, right? Bipolar neurons. We'll see them next semester when we talk about vision more. So let's look at a picture of them. I think that makes it a whole lot easier than describing them. So we'll start in the very middle, because I think it's the easiest one to characterize. There's the bipolar. What do we have? Cell body, two processes. One's called a dendrite, one's called an axon. Simple enough. So that's a bipolar neuron. That's its shape, its morphology. We won't be discussing them or seeing them this semester. There are multipolar, okay, down here on the bottom. Multipolar, here you see one axon and a number of dendrites. This one's just fancier. It has more dendrites, right? Lots and lots of dendrites, but it still only has one axon leaving the cell. So those are multipolar, and that's the shape, again, of your interneurons and the shape of your motor neurons. Yes?
Does it have to do with what it's receiving? Like, since sensory is only maybe coming from one area, it just...
It does. We're kind of, there's two things going on here, right? So I told you that neurons have different shapes and different functions. So you're kind of combining these two ideas. Yes, sensory neurons are receiving, but they also, by shape, are unipolar. Whereas motor neurons, which are sending instructions, oh, by the way, they are multipolar in shape. Okay, so I'm just kind of adding to the bigger idea of these neurons. So you're right. What I'm talking about, you said, does it have to do with their location or what they're doing? Absolutely. But, again, the sensory neurons are going to be unipolar. So let's go up to that one. Unipolar. Now look at this poor little cell. Looks kind of funny, doesn't it? There's your cell body, and it only has one pole, one process. Kind of goes against what we've already said, doesn't it? Because so far, all the neurons you've seen had what? Lots of dendrites, or one dendrite, and one axon. This guy, the process comes out and kind of comes to a letter T. And the dendrites are way over here, and all of this is considered axon. So it looks like I just told you a story, right? That I'm kind of lying to you. That I said, right, the axon always comes out and goes away from it. And here I'm telling you, oh, look, part of the axon comes in and part of the axon goes out. Don't let that trouble you too much. But why do you think they call this axon? What do you think is around all of this portion of the process? It's an axon because it has what around it? Yep, and Schwann cells, right? So it has Schwann cells around it, therefore it's going to be considered an axon. The dendrites are just these multiple processes over here on the end. Don't get too crazy about it, but we're going to see these unicellular shaped neurons quite often. So you'll be reminded of this quite a bit in the next few minutes.
So are those dendrites on the other end then?
Nope, on the other end, these would be what? Axon terminals, telodendria, or terminal arborizations. Good catch. So on this end, those branches are the ends of the axon. Information is still only going through this neuron in one direction. right? Information's only going in one direction. It just happens that the soma is kind of off to the side.
Is that just what keeps the cell alive, or does that have a function of, like, processing
The soma,
Yes.
absolutely, it's still processing. The soma is still processing. The soma is always where the nucleus is. It's still the center of the neuron.
So does it take a shortcut?
I know what you're thinking. Don't let it worry you too much. This is so much more complex than what I'm trying to get you to think about right now. So hang on there, and we'll see some more complexity build on this. So I've said these terms. You know what inter means. It means between. And you know that there are interneurons. Unfortunately, everything in anatomy has two names, and so do these. You'll see also the term association neurons. I, though, use the word interneuron more because it helps me remember what they are. Right? They're the between neurons. And these neurons are found only within the CNS. Right? They're only within the CNS. And so that's their location. Right? They're only in the CNS. And morphologically, they are multipolar in shape. Again, where are they? They're receiving the sensory neuron signals. They're integrating, processing, quote, thinking about, and then they're going to inform what? The interneurons are going to send their signals out, what? Out the motor neurons, right? Because these interneurons never leave the spinal cord. They never leave the brain. They're the little processors of the nervous system. So they're receiving the sensory information from the sensory neurons, they're integrating and processing it all, and then they're the ones sending the signals out via the motor neurons. In lab, I think I gave you a number of more than 99, I think I said in lab, I said between, what did I say? Over 90% of all of your neurons are interneurons. It's actually more than that. Here I'm giving you a more complete picture. 99% of all of the neurons in your body are actually interneurons. So that tells us that 99% of the nerve cells in your body are clustered inside your brain and inside your spinal cord. So let's put this whole thing together. So on the surface of your skin and all over your body, you have receptors that can pick up touch and pain and temperature and all sorts of things. So here in your skin, there are some receptors. These are sensory receptors. And let's say there's I don't know, a mosquito on your shoulder, okay? So you sense it through your sensory neurons. These are also called the afferent neurons, aren't they? So the afferent neuron is going to come in, and it's going to make it to your spinal cord. So this is your spinal cord over here. And it's going to come in, this portion of your nerve, we'll come back to this in a moment. Oh, look, what is this? What is that? What kind of neuron is that? What kind of shape is that? Unipolar, right? It's a sensory neuron, isn't it? So remember I told you your sensory neurons are going to be unipolar in their shape. So there we see it. Okay? Now this neuron is going to come in and is going to synapse. And what does that word synapse mean? Right? Syn meaning together, and APS meaning fit or fasten. So we're fastening cells together, if you will, very, very close proximity. And one cell, the sensory neuron, is going to synapse with, essentially come in contact with, the interneuron. The interneuron here is shown as purple. And what did I tell you about the shape of a interneuron? Which morphology does it belong to? Multipolar, right? Which means it has what? Many dendrites. Look at them all. Right? See all the dendrites? Only one axon. So this interneuron is going to do what? Receive this sensory information, process that information, and in this case, the interneurons are going to inform these motor neurons. Right? There's a motor neuron. What's its shape? It's also multipolar, isn't it? We see all the little dendrites. And it's going to tell that motor neuron to send a signal out of the spinal cord. Again, we call this efferent. And now that signal is going to go down to the end of the axon. These branches are called what? Axon terminals or telodendria or the terminal arborizations. And at the end of this whole thing, the axon is going to tell the muscle to do what? Contract. Squat the mosquito, right? Do something, dummy, right? It's biting you. It's going, it looks like it's going to the biceps brachii, right? The muscle of the arm. So even more specifically, what kind of neuron is this? Motor neuron? Yes, but one more term. If you go back and look at that table, somatic motor neuron, because it was what? Going to voluntary muscle. So because in this example, it's going to voluntary muscle, we would call that a somatic motor neuron. If instead, this was your heart, right? Or your gut, or a gland, then we would call it what kind of motor neuron? Autonomic, right? Autonomic. So that's how this whole thing kind of fits together. So we'll come back and look at this story over and over and over, and we'll keep adding more and more layers of intensity or detail to it. So that's the neuron story. There are also these other helper cells called the glial cells. And some books will call them instead the neuroglia. And we're going to find some neuroglia cells both in the CNS and in the PNS. These cells, as I said earlier, are smaller. Remember you saw all those little nuclei scattered around that one neuron? So you know that they're smaller cells, and they are capable of replacing themselves. That is, they are mitotic. They do not, however, send electrical signals. So again, they're the helper cells, the glue cells. They support and protect, but they're not involved with sending signals through them. And they far outnumber. And you saw that again, all those nuclei around that one neuron image. So a young adult would have upwards of 100 billion neurons, but would have up to 1 trillion glial cells, okay, within your brain and spinal cord. Pretty amazing, right? The number of cells squeezed into your brain and your spinal cord. There's so many of them, but they're small. So they're going to account for about half of the overall volume of your nervous system. So question, can you have a tumor that initiated from a neuron? Can a neuron get a mutation and start to grow uncontrollably? Why? They don't have centrioles. So you won't have a neuron-derived tumor, but you will hear about gliomas, right? So any of these other types of cells can go wacko and start growing out of control. So you hear about different brain tumors, and if you know somebody who's had a brain tumor or look at it again, you'll notice that those brain tumors are characterized by the type of cell that went crazy, right? And you'll hear about gliomas because it's one of the glia cells that is now uncontrollably growing, not the neuron, right? Because it can't. There's no mutation on the planet that's suddenly going to give a neuron a centriole. Not going to happen. So let's go through these cells. The first one, an astrocyte. When you hear the word astro, what comes to mind? Stars. For me, it's the Jetsons dog. But anyway, right? Stars. So if you're thinking stars, good for you. So these must be stars with what? Shaped like, or cells shaped like stars. Said it backwards, right? Stars shaped like cells, right? So what we're going to see is that these cells are the most abundant of all of them. So the astrocytes are abundant, AA. They are going to be responsible for maintaining the blood-brain barrier. You may know what the BBB is? not the Better Business Bureau, but the blood-brain barrier. Anybody heard of this? To protect your central nervous system. Pretty important, right? There is extra safeguards in place in your circulatory system. So when you consume medications or drugs or whatever, there's a greater stringency in those molecules getting into your brain tissue, because along the blood vessels and capillaries of your brain tissue, there's this extra level of protection. That's called the blood-brain barrier. Now, it's a good idea, right? So if you consume a toxin or some medication or some molecule that could potentially damage your brain, it's good that we don't let it in. But the blood-brain barrier also causes trouble for treating neurological disorders, because many medications which we want to get to the brain are stopped by the blood-brain barrier. So when treating brain cancers and when treating depression and some other types of neurological disorders, the blood-brain barrier creates an extra level of headaches, if you will, for the pharmacological companies, because they have to deal with getting the molecule over the BBB. Astrocytes are the cells that are going to maintain that blood-brain barrier. Astrocytes are also, because they're regulating what can go in and out of the blood flow and the brain, are also controlling your fluid levels of the brain. So they're really the gateway, right? They're really the sentries allowing molecules into or out of your brain. So they form this star-like structural scaffolding network. And when neurons are damaged, they're replaced not by other neurons, but by astrocytes. So astrocytes can divide, right? They have a metabolism. And as portions of the brain die off through age or damage, astrocytes are the cells that replace them by space. Now that space is no longer contributing to your electrical signals, right? So it's not a functional nervous system, but that's what replaces your damaged neurons. We also know that from a neuroscience standpoint, that astrocytes are helping direct neurons to their correct place. One of the most fascinating things about the nervous system is that you think every muscle cell in your body is connected to a neuron, and every cell in your body is in somehow, in direct connection to or in close proximity to a neuron. And how does that whole magnificent circuitry get put down and laid down properly, such that here we are walking and talking and functional? And we know that these astrocytes are sort of helping direct the movement as these axons, right, these long axons are migrating through the body to form connections. These astrocytes are helping to lead the way.
So is there like a issue with these, is that where hydroencephaly comes along? Does that play a part?
Hydroencephaly, water on the brain, these cells, yes, they would have a role in that, but honestly, I can't recall what that is right now, but that's a good connection. So any sort of water on the brain, any sort of edema of the brain potentially would involve these cells in some way or another. There'd be other mechanisms going on. It's not just a faulty astrocyte. Now, another one of these cells are the ependymal cells. Sounds a little bit like a what kind of cell? Epithelial. So these are basically epithelial cells, and they're lining the fluid-filled chambers of your brain and spinal cord. Tell me right now, what fluid do you have around your brain? Cerebrospinal fluid. We saw it around the spinal cord in lab. I mentioned it. It's also going around your brain. So guess what? These ependymal cells are lining your subarachnoid space where the CSF is found. And there's also these fluid-filled regions in your brain called ventricles that we'll talk about later. And these ependymal cells are there. In fact, these ependymal cells make the CSF. So that CSF fluid is coming from these ependymal cells. Then there are the microglia. Well, they must be what? Small glial cells, right? They're just little cells. They are the smallest of the cells in size. They're also the smallest in the number, the least numerous. And these cells wander around within your nervous system, and they're phagocytic. That means they can chew up. So these cells are going around and trying to destroy dead and dying cells and any sort of molecule or cell that would invade, if you were ever to get a bacteria up in your brain tissue or a virus, these cells would be going at the bit, trying to chop it up. And then finally, there are the oligodendrocytes. Let's break that word down. Cell, right, site, dendro, lots of dendro terms, right, in the brain. Dendro tree, oligo. Haven't gotten there yet in our vocab. It means just a few, okay, a limited number, a few. So what does this cell tell me? Break it down. It's a cell with a few tree-like branches, right? Oligodendrocyte. And what we're going to see is that oligodendrocytes make myelin. What other cells made myelin? Schwann cells, okay? Listen carefully. Oligodendrocytes are found only in the CNS. In fact, all of these 4 cells that I'm describing here, astrocytes, ependymal cells, microglia, and oligodendrocytes, these are the glial cells, as you see here, of the CNS. Okay, so by deduction, where must Schwann cells be? PNS, right? So the Schwann cells are only found in the peripheral nervous system. Let's take a look at a visual of these cells in this cartoon. So we're in the brain, right? It tells us up here we're in the brain. And let's start off with, I think, the most obvious one. What's this? This big old thing and this big old thing are those are neurons, right? Now let's look for the helper cells, the glial cells. Let's start off with the star-shaped cells. In this picture, they're green, right? So see this big old astrocyte here? And look what it's doing. It's creating a structural network, isn't it? It's got these long tentacles, if you will. But look what else it's doing. It's got its little processes wrapped around the capillaries, around the blood vessels going into the brain. Because I told you these cells were maintaining the blood-brain barrier, and they're maintaining the fluid levels in and out of the brain. This blue area down here in the very corner is of a ventricle. Now, a ventricle is a fluid-filled region in the brain. And if you look up here in this picture, you'll see those blue areas in the brain. And those are ventricles. They're fluid-filled. And that fluid is good old CSF. And so you'll see lining, right? Lining that fluid-filled region are the ependymal cells. So they're shown here in pink. They're basically epithelial cells, right? Lining the fluid-filled regions, making the CSF. Then, look up here. It looks like a little tiny neuron. Isn't it cute? But that's actually a microglial cell. There's a couple of them. There's another one over there. So those are kind of wandering around. Here's another one down here. They're kind of wandering around, and they're phagocytic. They're gobbling up things. The last cell type we haven't looked at is this one. That is an oligodendrocyte. And look what it's doing. There's the nucleus and the cell body of this cell. But what is it doing? It's reaching out with a few tree-like branches and wrapping what? Myelin around the axon. That's a very different looking architecture, isn't it, than a Schwann cell. What do we see about Schwann cells? Each individual Schwann cell wrapped around a discrete section of the axon individually and wholly. Here, the oligodendrocytes, one cell is reaching out with a limited or few number of branches and is now myelinating a section of the axons. So a different architecture. When you see this kind of oligodendrocyte, you know you're looking at the CNS, right? You know you're looking in the brain or the spinal cord. So I like this picture. So as far as pictures to label and make sure you know, I would definitely anticipate seeing this picture again because it shows 5 different types of cells and they're pretty easy to recognize.
Are the breaks in the myelin, are those called anything? Like
Nodes of Ranvier, same, right, same thing. Nodes of Ranvier, good. Those little breaks or those little branches, or not branches, breaks, right? Little gaps. There's what I'm looking for. Little gaps in between the oligodendrocyte sections are again called nodes around VA. So these are just individual pictures of what you've just seen. Again, if you look at this, you'll see the big old green star-shaped cell. It's got its, lots of structure to it, fills a lot of space, and wraps its little fingers around the capillaries. The ependymal cells lining the fluid-filled regions, making CSF.
Is that cilia on the outside of those cells?
Actually, it is. These would be cilia-like structures, absolutely. The microglia, very small. They look like miniature neurons in a way, right? They don't look much different than a neuron, except they're tiny compared to the big mama neuron right here next to it. And then here's that oligodendrocyte. One cell reaches out with a few limited branch-like structures to myelinate a section of a neuron, of an axon. What are those oligodendrocytes making? Myelin. What are Schwann cells making? Myelin. Myelin is really important. It'll make even more sense next semester when we discuss in 106 how action potentials and how electrical signals travel down an axon. But let's figure out what this myelin stuff is. Now, there's that word for you in big print. There's that word. Neurolemocyte, right? And what was that word again? What was that cell? Neurolemocytes were cells that did what? Rind or peel, right? Go around. So the Schwann cells are the same as the neurolemocytes. They're found only where? Yep, in the PNS. So associate PNS and Schwann cell and myelin together, please. Now, myelin, I told you, was a white, glossy, shiny, fatty-like protein that created an electrical insulation and a protective layer around that very important long thing called the axon. That's the only place you see the myelin, isn't it? If you go back and look at any picture of a neuron, you will not see myelin around the dendrites. You will not see myelin around the soma. You will only see myelin around the axon, okay? So you're not going to see it on the cell body. You're not going to see it up on the dendrites. So wherever you see, quote, white matter, when you hear about gray matter and white matter in the brain, white matter must therefore be what? It's white because of myelin. And myelin is only surrounding what structures of the neuron? Axons. So when you see white matter in the brain, we're dealing primarily with myelinated covered axons. What would gray matter be? If there isn't white, it's gray. What structures are making up gray matter of the brain? Cell bodies and dendrites. Okay, so I'll say this over and over and over, but if it's white matter in the brain, it's primarily myelinated axons. If we're looking at gray matter in the brain, gray matter is going to be the things that are not myelinated, that is cell bodies and dendrites. Question?
Why does it leave part of the axon uncovered and unprotected? Is there a reason?
There is. And I'm going to punt on that till next semester, or maybe I'll punt on that for about 5 minutes, and then we'll deal with that a lot more next semester. Yeah. There's a very important significance to the nodes around VA that we'll discuss. Now, I will say this, though. In the cartoons, it always makes the node around VA to look like a rather substantial gap, right? They really overemphasize the gap in between those Schwann cells. In reality, it's almost insignificant. So it looks like this big gap between the Schwann cells. It really isn't. Okay, but they always overdo it. So your idea is, why would we leave pieces of axons exposed? They really aren't. It just looks that way. Okay, but we'll come back to what's going on. So the myelin is protecting, supporting, insulating the axon. Now, look at this cartoon. Look at this orientation. What kind of neuron must this be? Give me 2 names to describe this neuron. Ah, I like it. Multipolar, right? Lots of dendrites. And another, what part of the nervous system is this guy associated with? PNS. Why? I see individual Schwann cells, right? I don't see that oligodendrocyte architecture. So I've only got individual Schwann cells wrapped around the axon. So I know that I'm looking at a neuron from the peripheral nervous system. I could call this a multipolar neuron. Therefore, this motor neuron is of what type must it be then? Ah. What kind of neurons were multipolar? Motor and interneurons. This can't be an interneuron, right? Because it's
In the PNS.
in the PNS. So this must be a what? A motor. Does everyone see where I just went with that? This has to be a motor neuron because it is multipolar and it has Schwann cells around it, which are only found in the PNS. It can't be a sensory neuron because sensory neurons are unipolar, and it can't be an interneuron because interneurons would have around them oligodendrocytes. Yes? Don't worry about satellite cells. So over here on the left, just cross that off. Not a concern this semester at all.
I have another dumb question.
There are no dumb questions. Just dumb answers from me.
They're really concerned about protecting the axon. Is that the way a cell would die or be damaged? Could it be damaged in the dendrites or on other parts? Would that, cause cell death?
If you damaged one dendrite out of 1,000, we got 999 others.
And they don't grow more.
They don't grow it back. In a few minutes, I'm going to talk about a limited opportunity for axons to regenerate. Not the entire neuron, but just the axon does have the ability of regenerating. But give me a couple minutes and we'll talk about those scenarios. Okay. We're almost there. Now, myelin, again, is this white, glossy protein that acts as an insulator. And this is going to assure, in part, that as that electrical signal is traveling down that very long axon, that it doesn't lose its charge. We don't want me to send signals down to my big toe, right, and the signal get lost somewhere around my knee. So we need to make sure that that signal makes it all the way down to the end, to the axon terminus. Again, I keep saying it, so man, I guess we better make sure we got it. Myelin is produced only by neurolemmacytes or Schwann cells in the PNS, whereas it's made by oligodendrocytes if we're discussing cells of the CNS. Let's take a look at a cartoon look at a Schwann cell. Okay, so here's a neurolemmacyte, a Schwann cell, and the yellow represents the axon. And what was the other name for an axon? I feel like one of those TV shows. I start writing letters, and at some point somebody yells out, nerve fiber, right? So nerve fiber, right? So axon equals a nerve fiber. So we're looking at a nerve fiber, right? AKA an axon. And here is a beautiful little Schwann cell that is beginning to wrap itself around the axon. Has its own nucleus, has its own metabolism. And what these cells do is they start to sort of wrap around and around and around and around. And eventually, you have layer after layer after layer of myelin. Hundreds of layers. This is kind of a simplified view. So you have this simplified view. And notice that there's very little cytoplasm in a Schwann cell. In fact, the nucleus gets shoved out to the outside edge. So whenever you look at a picture of a cartoon of this, you'll see the little purple nucleus kind of shoved to the outside edge of the Schwann cell, because really the Schwann cell is nothing more than hundreds of layers of myelin. Oops. So let's take a look at these two pictures. I think we have this now. This must be neurons from the CNS or PNS? Has to be the CNS because I see oligodendrocytes. Whereas this on the bottom must be a neuron from the PNS, because I see individual Schwann cells. We got it, right? No problem. We got that distinction. We won't forget that. All axons have Schwann cells protecting them. All axons, right? They're too important not to protect. However, you will hear it say that some axons are unmyelinated and others are myelinated. The difference here is not the absence or presence of a Schwann cell, but it is the difference in the way that the axon is protected. So let me tell you about this little story just for a little bit. And again, we're going to dive into this ad nauseum next semester. Someone asked, why do we leave these little gaps? All right? Why do we have little spaces in between the axon? Well, it turns out that the electrical signal travels more quickly and basically skips, like a rock skipping across a pond. And it kind of skips from node to node to node. Now, why that happens, we'll deal with next semester. But just know that the signal travels very, very quickly down an axon, and it essentially jumps from node to node to node. This jumping mechanism is very, very fast, and we call this type of conduction, this type of signal, saltatory. Saltatory conduction. So in essence, the nerve signal is jumping from node to node to node. Now, I never forget this term. When you hear the word salt, what comes to mind? You first think of sodium chloride, right? Table salt. And I'm going to tell you right now that sodium is very, very important in this process. Okay, so just remember that there's sodium involved and that it's saltatory conduction and it's jumping down the axon. Versus an unmyelinated axon. It still has a Schwann cell, but the Schwann cell does not wrap around it the same way. Well, I'll show you the picture in a moment, but what's going to happen is that the Schwann cell leaves a side of the axon exposed, which means that the signal up here, right, in saltatory, it jumped, right, from node to node to node. Because there are no nodes in unmyelinated axons, the signal must travel what we call continuously. So it can't jump. And what that does is it slows it down. So unmyelinated axons carry the nerve impulse more slowly than a myelinated axon. So again, if the axon is myelinated, if it has Schwann cells wrapping all the way around the axon, then it's going to speed up the speed of the nerve signal.
Is that just due to like surface area?
Part of it is surface area. Yep, it is. Now, going down an unmyelinated axon, again, it's going to take longer for the signal to get there. And I'll also say, though, that a myelinated axon is more efficient. It does not require as much energy as a myelinated axon, or as an unmyelinated axon, sorry, than an unmyelinated. It takes a lot of energy, right, takes a lot of energy, a lot of ATP for nerve signals to travel through or down an axon. Anybody know what disease we hear about way too often that involves myelin? What disease do we hear about that involves the breakdown of myelin? MS, multiple sclerosis. Anybody know anybody with MS? Okay. Way too common. We really don't know what's causing it, but we know that it's involved with the breakdown of myelin. So you hear about these people having demyelinating events or stressful events. And during those times, the disease is progressing and myelin becomes destroyed. Now, MS is a breakdown of the myelin of the central nervous system. So this is a breakdown of the myelin made by the oligodendrocytes. Okay, so something's going on and the myelin's getting destroyed. As the myelin gets destroyed, the nerve impulse starts to slow down, right? There's hiccups in the conduction of the electrical signal. So what sorts of symptoms does one with MS complain about? Numbness, tingling, and then later on maybe some more jerky movements. At some point they may have visual problems or speech problems, right, as the disease progresses. Some people can have MS for decades and just slowly progress and really do quite well for a very, very long time. Other people, for reasons we don't understand, have a much more rapid decline, and their demyelinating events, their myelin gets broken down more rapidly. We don't understand why, the differences between the two. Another thing that a person with MS will complain about is how tired they are, right? What am I telling you? That as you lose myelin, it takes more energy to do the same job. So the body is actually being robbed, if you will, of energy, of ATP, just to go about the everyday events of life. Okay, so that all kind of fits in if you know someone with MS. They're tired. They're tired. As the disease progresses, their body is just tired of working through all that extra energy needed to have motion and movement and fluidity in their body.
Is that part of the reason that it can't be cured is because the cells can't reproduce?
Well, absolutely. So myelin, we don't know what's causing the breakdown directly. And so far we haven't found a way of mending the myelin. Now there's another disease similar to MS, Guillain-Barré disease. If you've heard of Guillain-Barré, don't worry about it for notes, but there's another disease similar to MS, and Guillain-Barré is a breakdown of the myelin in peripheral nervous system, whereas MS is a breakdown of myelin in the central nervous system. So you may have heard of it, right? Guillain-Barré, similar, but yet different from MS. Now it's interesting that your pain signals travel along unmyelinated axons. You ever cut yourself? Burn yourself? Right? And the moment you burned yourself, your reflexes kicked in and you pulled your hand back, right? So you touch a stove and out, before you even said ouch, right? You pull your hand back. And then you go ouch, right? You ever done that? Right? You pulled away reflexively, and then you look at it, and then you go, man, that hurts. So the signal of pain traveled more slowly than the signal to pull away from the damage. So your pain signals travel through your body, largely on unmyelinated axons. That means that signal is traveling more slowly, and there's a fraction of a longer period of time from the time you injure yourself to the time where you feel or sense that pain. This is what an unmyelinated axon looks like. Okay? Here we have 1, 2, 3, 4, 5 nerve fibers, 5 axons. They're all being protected by one Schwann cell. But what do we see? It's protected, right? The axon is still protected, but it's not protected in quite the same way. And along one edge of the axon, what is there? There's still an exposed edge. So there is no gap. There is no node. So the signal can't jump from node to node to node. Instead, the signal must do what? Travel, quote, continuously down the axon. So it's slower, not as energy efficient. The neuron is still protected. Okay, so don't think, when you hear the word unmyelinated axon, don't think it's a naked axon. It's not a naked axon. It's not an axon without myelin protection, but it is an axon which has been designed not to be wrapped in the same way. This is not random, right? This is the way these neurons set up themselves.
Do they have a different function?
Well, they do. I mean, our pain sensations are slower, good or bad. Our pain sensations are slower. And so some of our signals simply are slower to travel through our body. Is pain important? Absolutely. But pain is not as important as your sense of sight or your sense of hearing or your sense of balance. So your signals for sight, your sensory signals for hearing, your sensory signals for balance, those have to be going at amazing speed, right? There's no way we could function if those signals were delayed in any way. But we can still function without pain being quite as fast. That's another way of thinking about it. You know, when you consider, okay, why is it that our visual neurons are myelinated? Zip, 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 right? Signals are going really, really fast. Whereas our pain neurons are unmyelinated. And you can kind of come up with some different scenarios to describe why the good Lord made us this way. Now, let's take a look at when can a neuron regenerate? The answer is never. But it is possible. You asked about what if a dendrite gets damaged? Big deal, right? There's usually many dendrites. But in this case, what if an axon gets damaged? Right? That's that one and only process. It is possible, limited, for a axon to regenerate. Now, once again, what kind of neuron is this? Multipolar, and it looks like it's going to muscle, so it's a motor neuron, and I see around it individual Schwann cells. So again, where are we? We're in the PNS, okay? So, in the peripheral nervous system, it is possible if you sever an axon, so here we are, right? This is a neuron. We'll see more of this in a minute, but this cell body is up in your spinal cord, and the axon is traveling down out one of your nerves and going to someplace in your body. And if you were to sever the axon, like out here, what happens? Well, the muscle is no longer getting its signal, is it? Therefore, that muscle can no longer contract. But it's possible for the body to have a little bit of repair here. So what happens is that all of the stuff down here, distal to the injury, will be destroyed, broken down. But as long as we keep these Schwann cells around, right, you see them? These Schwann cells basically are creating a tube. So we call this a regeneration tube, right? Because the Schwann cells weren't damaged, right? We just severed the axon. The Schwann cells down here are still viable. And as long as those Schwann cells are still down there, they'll basically create a path. Right? Was it Hansel and Gretel? Right? Little crumbs? It's basically that kind of idea. So these Schwann cells are kind of sending out little crumbs saying, come back home. And it will start drawing that axon back through the regeneration tube until such time that it can regenerate. You see it regenerating here, and eventually reconnecting to the muscle or the effector. Nancy?
How might that injury happen?
A crushing injury,
Okay.
okay? And let me just tell you that this is not automatic, okay? So let me tell you the parameters for this, and this will make total sense to you. If you were to cut, or even cut, right? Slice, cut, slice, crush, whatever, right? You damaged a portion of an axon. It can regenerate, not the neuron, right? We're not creating a new neuron. We're just extending the axon back to where it used to be. But there are some restrictions. And the restrictions on this process include the following. Number one, how much damage was there? Obviously, the more damage, the less likely you're going to maintain those neurolemmacytes to lead the pathway like Hansel and Gretel. Number two, do the neurolemmacytes stay around, and do they secrete a product called nerve growth factor? Now, nerve growth factor, as the name suggests, is a chemical which causes nerves to grow or causes the axon to regenerate. So if the Schwann cells aren't there, or if they're not producing this substance NGF, then it's not going to happen. The third thing is, what's the distance from the site of damage to the effector? So the example I always have is this. I have a 16-year-old son. About 2 years ago, he was working out in the gym, doing those big jumps up on a box type thing. And he landed funny and bruised the front of his tibialis anterior area. And he said, Dad, I can't feel that little area. I can't feel that little area. Well, he had a crushing injury. I mean, he had a really deep gash, and he clearly severed some of those axons. And I said, well, let's wait a little while. Let's just see if it comes back. And it never has, right? So he's never regained sensation. So either the injury was too extensive or the damage was too far away from where the muscle attached. So he just doesn't have any feeling in the front of his leg from that injury. Now, the rule of thumb here is that this can regenerate about a tenth of a millimeter a day. Okay, about a tenth of a millimeter a day is how long it takes for that regeneration to occur. So it's very, very slow. And if after 6 months it hasn't happened, it isn't going to happen. Now, the other example I always have is that every winter, being a Florida boy, I am not prepared for the snow. It's a surprise and a very big disappointment for me every single year. So I go out to my car sometime in December when that first snowfall comes down, and I'm out there wiping the ice and snow off my windshield without gloves. Right? So, okay, and I get back in my car and go, oh goodness, I can't feel my fingertips. So what have I just done? I've caused some sort of nerve damage right to my fingertips. And usually for the months of December, January, and February, I can't really feel it very well. But by March, April, when spring comes back, my fingers have thawed out too. So usually what I've done is, you do some nerve damage to your fingertips, frostbite type thing, and it usually does regenerate because the damage is so peripheral, it's so close to where the sensation goes back that it kind of regenerates pretty well. Question?
In my second pregnancy, I had Bell's palsy, and so I lost the whole left side of my face.
Okay.
I broke my tongue, all of it.
Right.
And I had some nerve damage, but it did grow back, but it attached to different muscles.
Yeah.
So now my smile is crooked, so it doesn't attach in the place that it was severed.
So Bell's palsy is a viral induced damage to the seventh nerve, the facial nerve. And suddenly you look in the mirror and you're,
Your
right?
food falls out
Yeah,
your mouth.
your mouth is droopy, you can't smile, and your eye, right, stayed open?
I had to tape it shut.
Yeah, you kind of, so to sleep, you got to tape your eye closed because it just stays open, you can't close it. And it usually recovers in, how many months do they tell you?
Say if not, by 6 months it doesn't.
Yeah,
Mine
so
was 6 weeks.
Usually, it's relatively short term and it will return itself, but you're saying that that damaged nerve did not go back quite to all of its proper homes, right? It got a little bit confused.
That's high smile on the one that's actually stronger, and then it goes to some extent.
Interesting, yeah. So I've never heard of that, but it makes sense to me that, now we don't understand, well, we just know we think it's a virus, right? All of a sudden you just wake up and a virus attacks the nerve, and it's interesting, it doesn't happen to other nerves, right? So Bell's palsy is a very limited, feature just to one side of the face, and it usually resolves. And I'm quite sure that in your textbook there's a picture of a person with Bell's palsy. Okay, so how are we doing? Any questions on neurons? Neurons, neurons, neurons. Okay, when I say the word neuron, and then I say the word nerve, there's a very distinctive difference here. So what is a neuron? What have we learned so far about a neuron? A neuron is a cell, right? It's an individual cell. It has dendrites, it has an axon, it sends its electrical information through it in one direction versus a nerve. A nerve, you name some nerves in lab. You name the femoral nerve, the median nerve, the brachial, right? The different nerves. A nerve's a pretty big structure, right? You can see it on the model. It's a pretty big bundle. So a nerve is a bundle of axons, right? So therefore, now let's think about that word fiber. A nerve is a bundle of nerve fibers. A nerve is a bundle of axons. And they're all traveling through the same part of the body. And these neurons, man, they don't travel by themselves, right? They're not just traveling, by themselves. They're traveling in big clusters called nerves. And they're traveling basically encased in a layer, encased structure. So it's a cable-like bundle of axons that are traveling in the same direction. Or I shouldn't say in the same direction, that are parallel to each other. You shouldn't be surprised, and these names should look really, really familiar to you. A nerve is made up of layers of connective tissue that protect these nerve fibers. And look at these names: endoneurium, perineurium, and epineurium. Looks a lot like the mesonems that we had in muscle. In fact, it's the same parallel idea. So if you didn't learn it then, here's your chance. And if you did learn it then, you're kicking back and saying, I got this. So endoneurium, remember endomesium was what? The layer of connective tissue around each and every muscle cell. So the endoneurium is the layer around each and every axon, fiber, right? The fiber. Then the perimesium was the layer around the fascicles. So too in a nerve, the axons run in bundles called fascicles, and around the fascicle there's the perineurium. Around the entire nerve, on the outside, epineurium. Right? So the same synonymous idea as what we saw in muscle. Let's take a look at a picture of this. So the entire thing is the nerve. This layer coming off the outside must be the epineurium, right around the entire nerve. Inside the nerve, I'm going to see groups, fascicles, and each fascicle is surrounded by perineurium. Within the fascicles, what's sticking up would be a single fiber, nerve fiber, axon, and that would be surrounded by a layer of endoneurium. And then even deep to that, then I would have my Schwann cell, and then the axon. Okay, so those are the layers. Now notice, what do these layers of connective tissue provide? A place for blood vessels, right? So just like in the muscle, those layers of connective tissue allowed for nerves and blood vessels to travel down into the muscle tissue. Same idea here. There are blood vessels traveling through your nerves. Don't forget, nerves are living cells. I mean, I forget it. I forget it all. I forget that nerves are made up of axons surrounded by Schwann cells or oligodendrocytes, and man, they need nutrients. So too, we need to get blood supply and oxygen to all the cells making up your body.
What are the white fibers that are running through each of the fascicles?
Connective tissue, right? So these structures in here, yeah, just layers of connective tissue. I wouldn't ask you to, well, no, I wouldn't have you label this. But what we're looking at here is a big piece of a nerve. So this would be a what? That's a fascicle, isn't it? See the fascicles? So that means that each fascicle around it, what would be around it? Perineurium, right? And then around each and every little fiber there would be endoneurium and the myelin. And then around the entire outside of the nerve there would be epi, right? We got that. If you're looking now at the second picture, you're looking at fibers. This is all part of a nerve. And if you look in here, what you're seeing are a bunch of long strings. Those are the axons. And then look right here. What do you see? See that little indentation? See that little indentation? So what we're looking at is what? That would be a nodeuron VA. So we're actually seeing the axon traveling along where my finger is, or along this area here that I highlight, and we can see that there's Schwann cells, and you can actually see that little cleavage, that little indentation, right, suggesting where the Schwann cell stops. So like I was saying, the cartoons always overemphasize the size of the node. In reality, there's very, very little space in between. So then what is a nerve? A nerve is something we find only in the peripheral nervous system. I'm going to introduce another term right now. It'll make more sense later on. And I think I said this in lab as well. A tract. A tract is essentially the same as a nerve. But a nerve is only found in the peripheral nervous system, whereas a tract is found where? In the CNS. So what you're seeing right now is that there are just different vocabularies, if you will, for things found within the CNS and things found within the PNS. Nerves can be sensory, no different. Nerves can be also motor, or they can be mixed. So again, what's a nerve? It's a bundle of axons traveling together. All of those axons could be sensory. They're all going in. All of those axons could be motor. They're all traveling out. But most of your nerves are going to be mixed. That is, that within that cable, within that nerve, some axons are traveling one direction, sensory, and neighboring axons are traveling the other direction, motor. The information is not going to be traveling up and down both directions in a neuron, right? The movement of electricity, the movement of signals in a neuron is unidirectional. But the movement of information within a nerve can be bidirectional, right? Because within that bundle, some are sensory, some are motor.
Sorry, did you say that they're both types in one fascicle? Is that what you're saying?
I didn't say that. So I didn't say that, but within a nerve, right? Think about those fascicles, good idea. And within the fascicle, just for now, imagine that every neuron in a fascicle was sensory. So that means that they're all traveling in one direction, right? Let's say they're all going into the body. And then there could be another fascicle filled with motor neurons that are traveling out of the body, but they're all wrapped up together within the same nerve. So the information highway going through a nerve can be bidirectional. But the information moving up and down a single neuron would only and always be only unidirectional. So what's a synapse? Where two cells come into close proximity, and we're talking here about two neurons coming into close proximity. So one neuron, second neuron. The first neuron we're going to call presynaptic, before the synapse. The other neuron we're going to call postsynaptic, after the synapse. Let's take a look at a picture of this idea of a synapse. Okay? So we're going to have two cells coming into close proximity. So here is one axon. Again, multipolar axon. One axon. And these down here are the what? Those little branches are the axon terminals or telodendria, right? And what do they do? At the very end of those telodendria, there are little synaptic knobs. And what do they synapse with? We normally think of axons coming in and synapsing with dendrites. But it's also possible for axons to come in and directly synapse with the soma. The real rule here is this. A synapse can happen anywhere where there isn't myelin. Right? Because myelin would block the ability of the cells to touch each other. So where do we usually not have myelin? Dendrites, cell bodies. So that's where we find synapses. Do you find myelin down here at the axon terminus? No, there's also no myelin at the very, very tippy ends of an axon either, is there? So that's where we have a synapse. In a schematic, you're going to see the picture above shown like this. So you have one cell body and its axon. We don't even bother showing the dendrites. Comes down, and this little Y represents the telodendria, synapsing to the next neuron, and then it going down. So this would be the presynaptic here, and the postsynaptic cell here. So let's take a look at how complex the nervous system can be. 3 different types of synapses. The one that I think along the way we've learned about are the axodendritic synapses. So this means that an axon is attaching to a dendrite. So here's a neuron, it's coming down, and the synapses are onto a dendrite, right? From an axon to a dendrite, axodendritic synapse. And I told you a moment ago, it's also possible for a neuron to come down and synapse directly onto the soma. That would be an axosomatic, axon to soma synapse. We're not going to discuss it this semester. We will next semester. And this one will kind of blow your mind a little bit. But I told you a synapse can happen anywhere where there isn't myelin. And oh, by the way, there's no myelin at axon terminals. And in your nervous system, there are axons that communicate with each other at the axon terminals, at the telodendria. Those are called axoaxonic synapses. Not going to talk about them, but I want you to realize our nervous system isn't just this little complex series of wires that follow one after another, that it's this huge web of neurons which all communicate with each other. Two different ways that synapses work. Those were locations of synapses, right? Axoaxonic, axodendritic, axosomatic. But there's two different types of synapses as far as function. The first are the electrical synapses. These are going to be more rare in your body. You're going to find smooth muscle and cardiac muscle has electrical synapses. For the purposes of right now, and for this unit, I want you thinking just about cardiac. It is true in smooth muscle as well. But we've talked about cardiac muscle a little bit. And what is it that cardiac muscle, you know to identify cardiac muscle with? What's the one structure? The intercalated discs. And intercalated discs, I told you in passing, were the way by which these cells were able to rapidly communicate one with another. The reason that when your heart contracts, all of the cells in the heart contract as a single unit is because your heart cells are connected through intercalated discs. And those intercalated discs provide an electrical synapse. That is, the electrical signal can go directly from one cell into the next. Zip, right? And it can go fast, 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 so that every cell is acting as a group, right? All the cells of the heart are acting in concert. So very, very important. Think of cardiac muscle when you think of electrical synapses. Most of the synapses, though, in your body are actually what we call chemical synapses, because there's a chemical involved in the transmission of the electrical signal. So again, this is the most common. And at a chemical synapse, a chemical is released. Those chemicals are generically called neurotransmitters. And the only neurotransmitter I'm going to introduce to you this semester is this molecule called acetylcholine, ACH. And acetylcholine is the neurotransmitter that controls all of the muscles in your body. So it's by far the most common neurotransmitter, and again, it's the only one we'll discuss. So let's take a look at and there are a bunch of other neurotransmitters, we're only going to talk about that one. Here's a picture of a electrical synapse. It says smooth muscle, that's okay, but it's also true in cardiac. I want you thinking about cardiac muscle here. And what you see is that one cell, this is the presynaptic cell on the left, is in close proximity in synapse with a postsynaptic cell. And the electrical signal literally goes zip right from one side to the other. The electrical signal goes right through. This is an electrical synapse. No use of chemicals. Very, very quick transmission of the signal. Versus in most cells, we have a chemical synapse. Coming down right here would be the axon terminus. Okay, a little tiny branch at the end of the axon. And at the end of the axon terminus, what do you have? A swollen area called a synaptic knob. So we're down at the synaptic knob level. The synaptic knob belongs to the presynaptic neuron. Down here is the next cell. This would be the postsynaptic neuron. In between, right, these cells come into very, very close proximity, and that's called the synaptic cleft, the little tiny space in between these cells. So what happens is that the nerve impulse travels down, and that nerve impulse instructs the neuron to release these little vesicles filled with acetylcholine. So those little blue dots are acetylcholine. They are the neurotransmitter, the chemical. And that acetylcholine then has to go across this little space and be picked up by a receptor molecule on the postsynaptic cell. So we go electrical signal to a chemical signal, and then back here to electrical once again. So what happens is that we go from electrical signal, we have to go through a chemical intermediate, and then back to a electrical signal. Takes a little bit longer, right? So electrical synapses are very, very fast. Chemical synapses are slower, still amazingly fast, but slower than electrical synapses. Okay? And we'll talk more about this whole synapse deal as we go further in the course. Really quick, what are neurotransmitters? Chemicals released from one cell, from the presynaptic cell, that bind to a receptor found on the postsynaptic cell. And this chemical release is going to initiate another electrical signal in the next cell. Neurotransmitters work in a unidirectional way, right? This is one neuron speaking to another neuron. The information is traveling in one direction, from pre to post cell. But how quickly can a signal travel down an axon? And there are two things that influence how quickly signals can travel down an axon or down a fiber. One has to do with the diameter of the axon. If you go back and look at that picture I showed you of a nerve, you might notice that inside, not every axon is of exactly the same diameter. Some axons literally are bigger than others. The bigger an axon, the more surface area it has. And the greater the surface area of the axon, the more quickly it can send signals. So a bigger axon can send faster signals, right? I always think bigger bandwidth, right? A bigger cable can send more information. It may not be completely accurate, but it works for me. A bigger hose can carry more water, okay? The second thing that speeds up or influences how quickly a signal can go down an axon is whether or not there's myelin there or not. We've already talked about that. If there's myelin there, wrapped around with nodes of Ranvier, then the signal is much, much faster. If instead it is wrapped around partially and there's a exposed edge to the axon along the entire length, then that is a slower rate of conduction. So a bigger myelinated axon is going to travel the fastest signal. And a skinny, naked, unmyelinated, right, axon is going to carry signals more slowly. Does that make sense? Okay. I'm kind of busting through this today, but I wanted to get through this lecture. I didn't give you a break, so I apologize. But a couple more slides here. And then I want to say the first slide of the next chapter, so I feel better, right? Just so I feel like I've gotten to the next chapter, but we won't go any further after the first slide, okay? I'll just feel better in my heart. So these neurons, right? I told you there's this amazing circuitry all over your brain, up and down your spinal cord. Amazing trillions of cells speaking with one another, billions of cells speaking with one another. And these neurons are arranged in a few different ways, circuit-like arrangements. So I'm only going to introduce to you two of them. And I want you thinking about these first two, converging and diverging. You can cross off the bottom two. Okay, not going to worry about those. Converging and diverging. So let me show you a picture of these and it'll make sense to you. So this first image is converging. I went too far. So converging, if you're all going to converge on the flagpole, what does it mean? You're all going to meet at the flagpole, right? So converging, I also said a converging muscle, like the pectoralis major, was a muscle where it all was like a triangle. So converging means that all of these neurons are sending their signals to a single or to a limited number of neurons. Right? So many neurons are speaking to one neuron. The signals are converging in to the spinal cord or into the brain. The other signal pathway that I want you to know are diverging. This is Facebook, right, or Twitter, right? So one friend tells his three friends, and then they all tell their friends, and before you know it, the whole world knows about it because the signal is diverging. It's going out and it's spreading very, very quickly. We'll talk about this when we get to chapter 14. But when I talk about fight or flight responses in the autonomic nervous system, I'll use the example when you're camping and the bear walks into the campground. What happens? Woo, right? Fear. And all systems are quickly put on full alert. right? Your eyes go big, you start sweating, you start running, your skin goes white, your heart starts beating, your respiration goes up. All that happens very, very quickly because those particular pathways in your body are divergent. So all systems are put on full alert very, very quickly. Okay, so that'd be one example to think about as this diverging system. Okay, so that brings us to the end of the first chapter. Like I said, I'm going to feel better if I just get to the first slide here and say that not chapter 13, this is going to be chapter 12. Again, my numbers are off. Chapter 12 is going to be all about the spinal cord. And we know that the spinal cord is a link between the brain and the rest of the body. But the spinal cord also has its own job. What's its one and only job? It is all by itself. The spinal cord is responsible for maintaining your reflexes, right? Reflexes don't have enough time for the brain to get involved. So reflexes are the sole job of the spinal cord. And I feel better. Okay? So we got to the very beginning of the spinal cord. Now, we'll discuss the rest of the spinal cord on Monday. That'll leave us talking about the brain on Wednesday, and I'm pretty sure I will be pre-recording that lecture on the autonomic nervous system. You have 4 mastering assignments, okay? And one on the nervous system, and you can go ahead and knock that one out over the weekend. And then, and I will duplicate those like I have in the past, so you can do them twice. And then there will be later one for the spinal cord, one for the brain, and finally one for the autonomic nervous system. So I will see you all very, very soon. Have a wonderful weekend.